Health is an eternal topic for mankind. We are born on Earth, and our health is intimately linked to the environment and ecology. The environment is the basis for human survival and development, and every subtle change in the environment will eventually have an impact on biodiversity, ecosystem, and our health. Exploring how the environment and ecology affect our health in the past, present, and future is the mission of Eco-Environment and Health. Eco-Environment and Health is a journal initiated by prestigious scientists with a strong editorial board, including multidisciplinary scientists from all over the world. With the support from EEH, you will have a better publication experience. EEH has its own offices of publishing, language, design, and broadcasting. The experts will accompany you like a harmonious band and spare no effort to sublimate your valuable scientific research into a magnificent and impactful masterpiece. We have a dedicated center for decision-making that deeply integrates informatics, ecology, and environmental science. It explores global research hotspots, predicts discipline trends, monitors the influence and dissemination of publications, and supports the journal's smart decision-making. Upon receiving your submission, our assistant editors will contact you immediately and keep you informed of the manuscript status throughout the entire process. The 14-day rapid peer review will make the publication of your research one step ahead. Your distinguished work is worthy of being known worldwide. We will promote your research globally in a three-dimensional manner through various social media platforms. In addition, the open access fee will be waived for the first 3 years, making your research easier to be read and cited by your peers. We continue to explore and innovate the service strategy of the journal. EEH leads ecology and environment to a healthy future.
Okay, so I'm going to take about 40 minutes or so and talk about some of the work that we've been doing over the last 5 or 6 years, really, on nanotechnology and agriculture, really with a focus on addressing this issue of global food, really global food insecurity, which is going to be a increasingly difficult problem for us to manage as we move forward. And then hopefully we'll have some time for some discussion and questions at the end. So I think the best place to start with this story is to kind of start, with our current perspective. And this is a paper that we published in 2019 in Nature Nanotechnology, and it was featured on the cover, which was really nice. And it basically makes the point that, the Green Revolution really dramatically increased agricultural productivity over the last 50 to 60 years, largely through the use of irrigation and agrochemicals. But what we've come to realize is that many of these conventional approaches really are not sustainable anymore. And there are a number of problems with our current agricultural system. And you can see one of these down on the right. Let me get my pointer here. This is the average annual increase in crop yield for 4 different crops. We actually have data for 11 of them. But the orange bars are 1975 to 84, and the blue bars are the more recent period of 2005 to 2014. And what you see is that the year-over-year increases of many crops is actually declining. So these are still increases, but the magnitude of the increase has been consistently decreasing, meaning we're becoming less efficient at producing food at a global scale. We also know we continue to have significant problems with hunger, and with 800 million people hungry every day, 2 million suffering from micronutrient deficiencies. And in addition to that, we know we have problems with the delivery efficiency of many agrochemicals, fertilizers and pesticides, often being as low as 1%. And this is an area where I think nanotechnology can really have an impact. But as I said, this is the big driver for our work and many people moving into this space right now. And this has been known for some time that given the global population that we're going to have by 2050, we're actually going to have to increase food production anywhere from 70 to 100%, depending on whose numbers you're using. And remember the slide we were just looking at, we're actually moving in the wrong direction and have been for some time. So we're going to have to turn that around, and we're going to have to turn it around in the face of a rapidly changing climate, a climate that's changing more rapidly than anticipated. And that is significantly challenging agriculture. We continue to lose arable soil for a variety of reasons. And then there are unforeseen factors such as COVID. So many of us have really come to the conclusion that we need another agricultural revolution. And this one really needs to change how we grow, distribute, and store food. So from the farm to the fork and beyond. And we really feel that nanotechnology can and will play a significant part in this. And our focus right now is really on this inefficiency, but many of our partners are working on other issues. So nanotechnology in agriculture is not a new concept. You can do a Web of Science search and you can find review papers written in 2002, 2003, although really much of the research in this area is more recent. But the approach is straightforward. How can we use nanotechnology to produce more food more quickly, or increase the efficiency by which we're using our resources, food, water, agrichemicals, or minimize or address the waste that's produced afterward? So these are just some of the papers from our group and others in the last few years. The strategy is pretty straightforward. You just put nano in front of whatever term you're talking about and move forward. So the research at the Agricultural Experiment Station really comes in two separate tracks. The applications work is what I'm going to talk about today, our nano-enabled agriculture. But my training at Cornell was actually as an environmental toxicologist. And when I started hearing about engineered nanomaterials in 2005, 2006, I really viewed these as the next emerging contaminants, the next PFAS or the next microplastics. So I established a research program in nanotoxicology, looking at how these materials were going to negatively impact plants and agricultural systems, because I work at an agricultural experiment station. And I still have some work in this space, but what we noticed after a couple of years was that, not only were there instances where nanomaterials were not toxic, but they actually conveyed benefit to the plant. And that's when this more recent applications-driven program started. And these are some of the areas that I'll touch upon today, so I'm not going to mention them now. But a lot of this work is funded by USDA, United States Department of Agriculture, but I'm also part of two centers, and I'll present some of that data. But I think the important part here are these red arrows that I'm showing, because being trained as an environmental toxicologist, my perspective on this is if we can't sustainably and safely increase food production with nanotechnology, then we shouldn't be doing it. So these application and implication driven programs are not mutually exclusive. In fact, it's the exact opposite. They feed into each other. So our work in the applications area, nano-enabled agriculture, really began around 2014. We started working on some soil-borne diseases. We have a number of plant pathologists here who really have some very useful systems to work on. And soil-borne diseases are really difficult to manage. You pick a crop and you're going to lose 20% to some form of soil-borne disease, whether it's a fungi or a nematode. In the United States alone, we spend about $600 million on control efforts with fungicides, and we still lose $200 million in yield every year. I prefer to think about this in terms of lost food, though. So we know that many micronutrients, copper, manganese, zinc, we could list 11 or 12 here, they're part of plant defense systems, or I'd like to think of it as almost plant immunity. And when a plant's healthy, they can generally get enough of these nutrients from soil or from what the grower is supplying. But as soon as that plant becomes stressed, the availability of these nutrients becomes very, very limited. Plants can't get enough. So we know that if we add these nutrients to soil, they very rapidly become unavailable. For soil chemistry reasons we don't need to worry about. But we also know that when we apply them to the leaves, they're not really systemically transported through the plant. And if you're talking about soil-borne diseases, you need these nutrients in the roots. So we asked a very simple question: what if we started adding these micronutrients in nanoscale form? Could we effectively enhance nutrition, stimulate immune response from the plant, and really suppress disease? So we're not targeting the pathogen itself in terms of pathotoxicity. We're actually targeting plant nutrition, which is seems like a subtle difference, but it ends up being really, really important. So this was our first paper we published in this area. We published it in EEH Nano in 2016. The approach was really straightforward. Greenhouse and field trials with 2 plants that we had good disease systems with, eggplant and tomato. We used commercially available nanoparticles at the time. We used 3 nutrients, micronutrients, copper, manganese, and zinc oxides, but we compared them to their conventional forms, the bulk and the salt form. So we did a single foliar application to seedlings, because this is how a lot of agriculture happens in New England. The seedlings are started in the greenhouse and then transplanted to the field. So it's a foliar amendment to the seedling. So even though we're doing 100 ppm, we're actually only adding 1 to 2 milliliters because of the size of the plants. And then we transplant either in a greenhouse or field and grow them till harvest. So this is probably too much data, but what you see over here, and this is the field data for the eggplants. The green is copper, which performed best here and typically performs best. And this is our field data. So this is the number of fruit per plot. This is disease rating, and this is the number, the amount of copper in the root system. And the middle bar is actually the nanoscale treatment. So what you see is that single foliar application to the seedling is conveying benefit throughout the entire life cycle of the plant, whether you're measuring fruit, whether you're fruit produced, whether you're measuring the amount of disease. You're getting a significant reduction in disease here with that nanoscale copper, and you're getting actually more copper in the root system. So, kind of really proving our hypothesis. And when you do an in vitro test with this amount of copper, it's actually not even toxic to the Fusarium pathogen in vitro, because the amount we're adding is so little. But this was what really got us going and got our first USDA grant. Because this is a field study, we could do these calculations. We spent $44 per acre on nanoscale copper. And what did we really do? We took a nutrient that the plant needs, can't get enough of, and we just supplied it in a unique form. So that $44 investment actually led to about a $10,000 increase in economic return, or again, the way I like to think of it is in terms of food production. So at that point, we actually became part of an NSF chemistry center called the Center for Sustainable Nanotechnology. It's a 10-year center. We're just about ready to start our 8th year. It's a chemistry center, so it's really focused on the chemistry of the materials and what's happening in that leaf biointerface as we apply these materials. So here we compared commercial nanoparticles to a custom synthesized copper phosphate. We wanted to see if we could optimize behavior by changing the morphology of these materials. We changed the nanosheets and the composition. And what you see is when you do that, you dramatically change its dissolution profile and how it sits on the leaf. So this is a paper we published in 2018. This is greenhouse data with watermelon and again, Fusarium. We measured concentrations from 10 to 100 ppm, but the same approach, foliar treatment of seedlings. This is a measure of disease. So here's your control. Disease is really high. The yellow bars represent the commercial copper oxide, just like the previous study. And again, you got to get up to a couple of 100 ppm addition to see a decrease in disease, or the yellow bars here to see an increase in the biomass of the plant. But look what happens when you control or you tune the chemistry of your material. You're able to significantly reduce disease with these copper phosphate nanosheets at 10 ppm. That's about 10 times lower than the commercial material. And you're seeing that equivalent increase in biomass. So the real point here was by tuning the chemistry, you can really optimize your impact. And again, if you're worried about risk, which we always are, this is a very good approach. So this was a paper we published in 2020, in December 2020, on again in Nature. Nanotechnology. We got the cover on this one as well, which was very exciting. This was work carried out by my postdoc, Xuanzhen Ma. And here we really did a deep dive on the mechanisms. We compared several different types of copper nanomaterials. We switched to soybean, which is a much more significant crop, and the pathogen that causes soybean sudden death syndrome. In our greenhouse studies, this pathogen, a fungal pathogen, was reducing biomass and photosynthesis by up to 70%, causing all sorts of problems in terms of antioxidant enzyme activity and other things like fatty acid profiles. But our single foliar application of these different copper nanomaterials alleviated much of that damage. And here we looked at the gene expression of almost 24 different defense and health related genes. And we were really able to tie this observable phenotypic benefit in the plant to this stimulation of plant defense, specifically with nanoscale copper. You don't see it with the conventional forms. And we were able to tie the level of benefit to the different properties of the different copper nanomaterials. And that we were able to actually model computationally, which is probably what got us into nature nanotechnology. So again, the point of this is you can really exert tremendous impact on your final end benefit by tuning the chemistry and understand the mechanisms of what's going on. This was a paper that we published earlier this year in Journal of Agricultural Food Chemistry. We're specifically trying to really understand the chemistry at that leaf interface when we're applying these materials. So we took a step back and we started using some Arabidopsis mutants that either had increased or decreased stomatal activity or increased or decreased cuticle thickness, because we really wanted to figure out how these different types of copper were attaching to the leaf surface and moving through it. We tracked this from 15 minutes to 8 hours, looking at the different fractions of the leaf, so we could actually remove the easily removable fraction of copper, so loosely attached. We could isolate the cuticle itself, just the cuticle, and then look in the whole leaf. So we tracked all of that from 15 minutes to 8 hours with the different forms of copper. And we noticed some interesting things. So when you add conventional copper sulfate, what you actually see is that the amount of copper that you detect by ICP is 7-fold greater in the cuticle than with the other nanoscale forms. So this is really interesting because when you add copper in the conventional form, 7-fold more is getting stuck outside of the leaf in the cuticle, whereas the nanoscale forms are moving in much more quickly. And we've noticed this with multiple plant species. This happens to be with Arabidopsis, but we've seen it with tomato as well. And it turns out the cuticle was not important to the amount of copper that was coming in. It was actually all about the stomata, which is not surprising. I think others have shown this now. So if you've got increased stomatal activity, you get increased copper. If you get decreased stomatal activity, you get less copper in the leaf. And you can actually control that with abscisic acid, which will physiologically cause the stomata to close. So one of the other materials that we're working a lot with is silicon, specifically mesoporous silica in different forms. This is a very similar approach. This happens to be mesoporous silica that was coated with chitosan that we produced in the center. We looked at fusarium and watermelon again as a system. We did actually a seed treatment as well as foliar application similar to the copper work. With the seed treatment, you do get increased silicon content in, and you don't see that with the silicic acid, and that promotes germination and suppressed disease. What was interesting is when we looked at many of these stress-related genes, these are the same genes we were looking at with copper, instead of seeing an upregulation with treatment and disease, we actually see a downregulation. And that's due to the different mechanism here. And what's happening here is the mesoporous silica is actually stimulating excess deposition of cell wall material in the roots. So you're creating a better barrier. So actually less pathogen is making it in, which is why you're seeing overall less stress. So it's a different mechanism from copper, but the end result is the same. So we also did a field experiment. And as often happens, when something works in the greenhouse and you take it out in the field, it completely doesn't work at all. And our disease system in the field with watermelon, our treatments had no impact. But what was interesting is under the healthy conditions, that's actually where we observed a significant impact. We actually had a 70% increase in watermelon fruit production. And again, that's from a 1 to 2 milliliter application of a 500 ppm solution. So that's 0.5 milligram per plant when they're a couple of weeks old. And months later, we're getting about a 20,000 pound increase per acre in food produced from that simple $19 per acre investment in silicon. And again, that's just a nutrient that the plant needs, equivalent to almost $8,000. So this paper was published in 2019. This paper was published in 2021. This was a follow-up study with silica where we really wanted to see how important dissolution was. So one of the PhD students at the University of Minnesota synthesized 5 different kinds of silica nanoparticles that actually were physically and chemically identical to each other with the exception of how rapidly they would dissolve. So it was a function of these hydrolysis rates. So we set up a similar experiment. In this case, we're doing 1.8 mil of 1,500 milligrams. But again, it's just a couple of milligrams per plant. And this is too much data, but when, the take-home message was that of those 5 different types of silica, the one that performed the best, the one that gave us the greatest suppression of disease and increase in biomass and increase in yield, because this is a field study, was actually the material that dissolved the most quickly, released the most silicic acid the most rapidly. But what's interesting is if you just foliar apply silicic acid, don't go nano at all, just supply the silicic acid, you see no benefit at all. So even though the most rapidly dissolving material was best, there's still something very nanoscale specific about this benefit. And then from a scientific point of view, that's a really interesting question that we're trying to answer. But again, we've got significant increases in yield at field scale with these materials. So one of the other centers I'm involved in is from Singapore. Singapore funded the Harvard School of Public Health, where I had an appointment. And we had a project focused specifically on two aspects. This is the first one, where we were looking at seed treatments. Now, seed treatments are not new. They're very commonly used to deliver a whole range of materials and convey benefit. But when we looked at the current platforms, they're kind of limited in terms of what they can do. So this was work done with Phil Democritou, who actually just moved to Rutgers. And what we did was we used electrospinning to develop a biodegradable biopolymer-based seed coating. And the seed coating is just the carrier. So it's electrospinning. So these are nanoscale fibers. You can load anything you want in these fibers. And the idea being to get precision delivery, the right material at the right place at the right time. And a 4th R would be the right dose. So we tested this with lettuce and tomato in the greenhouse in the presence or absence of fusarium. This is just showing you the chemistry of these materials. We published this in 2020. I'm glossing over about 8 or 9 months worth of method development here. But the idea being you could electrospin these biopolymer fibers and you can onto the seeds. You can vary the recipe, and what you do is you vary the thickness of the fibers and how quickly they will release material. So this concept of tunability becomes really important again. In this case, we just loaded copper, not nanoscale copper, just generic conventional copper as our cargo. But again, you could load anything. So we created 3 different seed coatings that would release copper at a fast rate, a slow rate, or a biphasic rate. The biphasic rate was the fast layer on the outside, the slow layer on the inside of the coating. So this is just the time to germination data, which is a critical endpoint. So here you have tomato and lettuce under healthy conditions. This is tomato and lettuce under disease conditions. The green bars represent our coatings. And for this type of experiment, you actually have to have a lot of different types of controls, including we have some conventional film coatings here. But what we were able to see was that so this is time to germination, so days, so a lower bar is better. You're getting more rapid germination. And for the healthy tomato, for example, we were able to decrease time to germination by 22%. But what's really interesting is when we looked at the disease system, the presence of the pathogen actually adds almost 2 full days to time to germination. But if you're doing these seed coatings, whether it's fast, slow, or biphasic, and in the case of the lettuce, it was the slow that performed best, you can completely overcome the reduction in germination rate that the pathogen is causing by using these seed coatings. And I'm not showing you biomass data because we don't have time, but increased rates of germination obviously lead to larger and healthier plants. So this is a paper we also published in 2020. And this is where I started to get interested in a new material that we're working with, and that's sulfur. But this study was actually done by a PhD student at the University of Massachusetts. I was on her committee looking specifically at copper sulfide. We know what copper does, and we know why it does it for the most part. But sulfur we're also fairly interested in because 1, it's a required nutrient, 2, it's antimicrobial, and 3, it's tied into abiotic stress tolerance in plants because of the glutathione pathway. So there is some tunability here as well. The PhD students synthesized copper sulfide at different ratios. We're working with rice and pecane disease, which is a fungal pathogen. It's a greenhouse study. We did seed treatments and foliar, measured these things over 21 days with a variety of endpoints. This is just giving you the properties of the material. But the take-home message is this stuff really worked. This over here is our disease incident data. So here's your seed treatment and foliar application. So this green bar is healthy control. There's your disease control. And what you see is our copper sulfides are significantly suppressing disease, and they're doing so to a greater extent. That's Coside. That's a commercial fungicide, although it's not listed as seed treatment. But our copper sulfides are outperforming, whether it's seed treatment, foliar application wasn't quite as strong, but they're outperforming conventional pesticides. So that study really started to pique our interest in sulfur. I actually had a colleague at UMass Amherst who had been trying for years to get me to work on sulfur, and I really just wasn't all that interested in it, but that previous study really piqued our interest. So we submitted a USDA grant and got funded, and this was the first paper that was published out of that study. We're specifically looking at nanoscale sulfur. This was published in ACS Nano a couple of months ago to suppress disease in tomato. And here we use pristine nanosulfur, but also a stearic acid coated material. And sulfur is an interesting material because in the United States, farmers didn't worry about sulfur 20 or 30 years ago. But because of the Clean Air Act and less burning of fossil fuels, there's been less sulfur in precipitation, and actually we're seeing sulfur deficiency in soils. So many growers are actually adding sulfur to soil just as part of their normal fertilization regime. The point of me saying that is, in this experiment, we actually looked at nanoscale sulfur as a soil amendment because we wanted to model what growers are actually doing. So we used bulk and ionic as the controls, conventional controls. And our postdoc at the time, Yi Wang, who we since hired as a staff scientist, really measured a large number of endpoints here, everything from agronomic parameters to gene expression and metabolomics. And we did this over it was a time-dependent analysis. Plants were harvested at specific points. So the presence of disease, and this is a greenhouse study, the presence of disease decreased biomass by 87%. But our sulfur treatments, as you can see here, this is the measure of disease. Our sulfur treatments are significantly suppressing disease. Now the bulk and the ionic sulfur do okay, but they actually exert other negative impacts in soil that nanoscale sulfur doesn't. As I mentioned, this is time dependent. We harvested plants at 4, 8, and 16 days, and you can see the increase in biomass over time and that it's nanoscale specific. Interestingly, we had some 2-photon microscopy and time-dependent gene expression, all of which are demonstrating that it's actually the nanoparticle form of sulfur that's moving in through these plants. We were able to demonstrate that by orthogonal techniques. You don't often see that with other materials, but with sulfur, the particle itself seems to be really important because the traditional sulfate pathway, which most plants use, was not being activated from a gene expression or metabolomic point of view. It was this other elemental sulfur pathway, which is much less common. But the really interesting stuff was the time-dependent metabolomics, which lined up well with the gene expression data. And we were really able to show something that we had kind of seen in other studies that there's a really important time-dependent window here. If you want to stimulate a plant's immune system, you really need to do it very early in the life cycle. We've seen before in accidental studies where we didn't do the treatments early enough, where the benefit was lost. It really needs to be in that first 2 to 3 weeks. And we see that with the gene expression. As you go from 4 to 18 days, or excuse me, as you go from 4 to 8 days, the expression of these defense genes all increases, but then it starts to decrease at 16 days. But when you look at the metabolomic data, it's low at 14 days, it starts to increase at 8 days, and by 16, everything is high. So this physiological time-dependent window is really, really interesting. We're trying to figure that out right now. So that was part one of that sulfur work. We also did a field study, which is currently under review at JAG Food Chem, should be accepted shortly. It was a parallel experimental design, all the same treatments. And in this case, what we actually did was only measured yield and then the ICP content of the fruit. So in terms of ICP content, one of the interesting things that we noted was that we seem to be getting a biofortification of specific micronutrients when we're adding sulfur. The other thing I should mention is we also included one foliar treatment in here with the stearic acid coated material. But on a per acre basis, you know, our sulfur is costing us about $33, even as a soil amendment. And we were able to increase the marketable yield from about 5 kilograms up to nearly 12 for the healthy treatments. And for the disease treatments, from about 1.1 kilogram up to 2.1. So 2 to 3-fold increase in yield, equivalent to $6,000 to $12,000 per acre, depending on which treatment you were looking at. So this work with sulfur is really proving to be very, very interesting. So another project that we have that just started, this is a USDA funded project that started January of this year that one of our scientists is working on, a virologist. He's working specifically on plant viruses. Now, gene silencing is kind of the plant's evolved response to viral infection. So when a plant senses foreign DNA, it produces these enzymes that chop up that foreign DNA. Pretty straightforward. So you can actually trigger this response by supplying foreign DNA to the plant. The idea again being to stimulate the immune system so that when the plant actually encounters the pathogen, it can defend itself. The problem with that is genetic material is obviously very unstable and it breaks down quickly. So the concept behind this grant, and we don't have any data yet because we just started, is to actually use nanoscale carriers for that genetic material. So you can not only prolong how stable it is in and on the plant, but you can actually control its release. So you can control the plant's response to this virus. So this is work that's being done in collaboration with the University of Minnesota. We published a couple of papers that present the concept on this as well. So all of that, which I've discussed just now, is on biotic stress. We're also looking at how plants respond to abiotic stress. And this is work that was done by Chris Dimka, who joined us in 2020. And he's been interested in zinc oxide for a number of years. So this was a study that was done and was published in 2019. It was done with sorghum and specifically looking at sorghum grown under drought conditions. So in this case, the D doesn't mean disease, the D means drought. But this is yield, nitrogen, and potassium data. Here is your healthy control. Here, this is the drought control. And obviously under drought conditions, you see a decrease in yield and other nutrients. What's interesting is when you do relatively low zinc oxide amendments to soil, you can alleviate much of the damage that's occurring by that drought. And there's a couple potential mechanisms that we're looking at here. You also increase nutrient uptake, which leads us back to this concept of biofortification. So we've done some follow-up studies really trying to figure out the mechanisms of this. And Chris actually just got 2 USDA grants last year and is hiring a couple of postdocs to work on these types of projects. Another abiotic stress that we're interested in is salinity. This was work that was done under the Center for Sustainable Nanotechnology again. Our collaborator at UC Riverside actually was very interested in cotton. So this is probably one of the few studies I'll show you that's not on a food crop. But it's a seed treatment with a coated cerium oxide nanoparticle. Now, cerium is actually not a plant nutrient. But because it mimics plant hormone activity and it's a ROS, reactive oxygen species scavenger, when you supply it to plants, they tend to do better. And that's exactly what we saw here. So we did a seed treatment. We got increased cerium in the seed, in various tissues in the seed. And that priming increased the growth and vigor of the plant, including in the presence of salinity stress here. So we did some modest gene expression analysis. And again, a lot of it focused, a lot of the enhanced activity was related to ion homeostasis and reactive oxygen species pathways. By stimulating those ROS scavenging enzymes, you're able to handle the stress and get better growth under marginal conditions, which is going to be really important in agriculture in the future. So we're also doing some work on some non-stress systems, and you can't really talk about the unsustainability of agriculture without talking about phosphorus and nitrogen. Nitrogen's really difficult, so we decided to take a run at phosphorus first. This is a USDA grant. We're in our second year. There are several issues with phosphorus, but a lot of it's focused on the fact that when a grower adds phosphorus to the soil, only about 10 to 20% ever makes it to the plant. The rest either becomes unavailable or it's lost as runoff, which causes all sorts of ecological damage. So the concept here was, well, let's take all of the phosphorus that the plant needs and let's embed it in a biopolymer, a polymer that is made of a material that's actually produced by bacteria. So then you can add that phosphorus in the polymer to the soil, and then the phosphorus will only be released as the polymer is degraded. And again, back to this concept of tunability, you can use different types of polymers, you can use polymer blends, you can use different types of phosphorus, and you can really control the rate at which the phosphorus is coming out of this material as the plant needs it. So it's not becoming unavailable in soil, it's not being lost. At least that was our concept. Here's our first paper in this area. We published it at the end of 2021. It was a 150-day study with tomatoes in the greenhouse, full life cycle study. And we measured a whole range of parameters and basically showed that when you add phosphorus this way, added in a polymer form, you get statistically equivalent plants and fruit compared to the conventional strategy, which was really important. You don't want to get different results in terms of, fruit nutrient content or something like that. So we got equivalent plants, but we measured leachate, which is not runoff, but it's our measure of runoff. And what you see here, this line here, these triangles, this is the amount of phosphorus that's leaching out of these pots over 150 days when you add phosphorus in conventional form. This is the amount of phosphorus that's leaching out when you add it in a polymer form. So you're getting about an 80 to 90% reduction in loss of phosphorus. So that implies a couple of things. One, maybe you can just add a lot less because you're keeping it all in the soil, or two, maybe the next growing season you don't have to add as much. So we've got some follow-up studies, including a field experiment going right now looking at some of these materials. This is a really interesting study that we just published a couple of months ago. This is back to the center with Harvard and now Rutgers in Singapore. We're visiting Singapore later in October. But this is getting to a platform that I think is going to be really important, and that's responsive nanomaterials. So nanomaterials that contain agrochemicals, and they release those agrochemicals in response to a very specific stimuli. So again, this is electrospray with biopolymers, and this is a core shell approach. So different polymers in the core and the shell, and then we can load different materials, again, in the core or the shell. So you can really control a lot of the chemistry here. What we loaded was actually nitrogen, phosphorus, and potassium, as well as copper. We loaded it into our biopolymer, we called them nanocapsules, at 100% of the label rate of these nutrients, but also at 25%. Thinking that if we could deliver these more precisely and effectively, we actually could reduce what we're adding by 75%. So the shell, the outer portion of this capsule contains chitosan. So it's pH sensitive, meaning as the pH lowers, as a root grows close to this capsule, that decreased pH will cause the core to break, the shell to break apart and release its material. And then the core was actually enzyme responsive. We made it responsive to a specific fungal enzyme, thinking if a pathogen was present, then you'd get the release of this. But again, it was NPK. And again, you can control the distribution of the NPK in those particles as well. So it's pretty large matrix. So much of this paper, which was published in ACS Nano, was actually on the chemistry of the material and how it behaves. But we did do a plant study with wheat and soybean. This is just showing you the photosynthetic data. And I know I'm running short on time, so I just wanted to focus on, if you look at this, so this is relative chlorophyll content and photosynthetic output. But what you see for many of these measures is that our nanostructures at 25% NPK and copper are performing equivalent to the 100% conventional strategy. So we're getting equivalent photosynthetic performance with 75% nitrogen, phosphorus, potassium, and copper, which is a pretty significant discovery. We also are getting some biofortification, which we're trying to figure out as well. We have a USDA grant in to expand some of this work. So this is actually some work that we've been doing with Li Xuan at Nanjing University. This was the first paper that she published in JAG Food Chem in 2020. And the idea here is, can we enhance photosynthesis? Many of these materials, things like iron and molybdenum, for a variety of reasons, they will interact with electrons or facilitate electron capture or facilitate electron transport. So the concept is, can you add, and they're all nutrients. So can you do a foliar application with these and enhance photosynthesis? Because if you enhance photosynthesis, you enhance carbon fixation, you enhance biomass and yield. So this was the first study that she published, and it was actually trying to develop a high throughput system so we could rapidly test materials. So it was leaf mesophyll protoplasts. And this is quantum yield that you see here. The black bar, the black line is the control. And what you can see is the green and the red are manganese oxide and iron. They are increasing quantum yield in this protoplast system. There's a number of other endpoints you can measure: ATP production, carbon fixation, things like that. We included silver as a kind of a toxic control. And what you see is that many of these endpoints, here's ATP production, are actually significantly increased when you supply very low quantities of these nutrients, which are known to either be semiconductive or to interact with electron transport system. So obviously a high throughput system doesn't do anything if it doesn't actually predict what a whole plant will do. So she included some whole spinach plant studies, and the short of the long was these materials also in an in vivo study significantly increased all the photosynthetic parameters, including or as well as biomass production. So we actually just submitted a really big USDA grant in August to expand some of this work. This was a paper that Li Xuan also we published together, although she did all the work, looking at molybdenum disulfide, and this actually informed the choice of plants in that grant that we just produced, because she took a step back and started working with a single-celled cyanobacteria, but a nitrogen fixer, because the concept is, if you're upregulating carbon cycling, and you look at an organism that also fixes its own nitrogen, what happens to that nitrogen cycle when the carbon cycle is upregulated? And the answer, which she saw in this study, which was also published in ACS Nano, was that essentially the nitrogen cycling will increase as well, for no reason other than to keep up with the carbon cycling. So this really got us thinking about things like soybean and alfalfa. So if you could stimulate photosynthesis by a foliar amendment of a micronutrient, you could also stimulate the rates of nitrogen fixation and nitrogen cycling. So I think I'm hopeful we're going to get this proposal funded and be able to do some really interesting work. So just a couple other concepts I wanted to touch upon. What's the technological readiness of all of this? So this was a workshop that we held in 2019 at McGill University. The premise of the workshop was that nano-enabled agriculture has tremendous potential. So we wanted to do two things. One, we wanted to look at all of the potential applications, And we wanted to rate them in terms of performance level and readiness level, which was interesting. But the really interesting part of this workshop was we wanted to identify the barriers. What was going to prevent us from taking nanotechnology into the field to start to address this food security issue? And we identified 3. And the interesting thing is only one of them had anything to do with science. Well, not anything, but only one of them is directly related to science. Sorry. And that was field scale demonstrations. And we've been spending a lot of time working on this. We published this paper in the first issue in Nature Food. But these other 2 issues are still proving to be problematic, and that's regulatory and safety concerns and consumer acceptance. So the important part of this paper was we actually identified strategies for overcoming all of these barriers. So it's a really interesting paper if you want to take a look. This is a more recent look. This was just published in Nature Nanotechnology. I think it was the April issue. Where a postdoc who's now at Auburn University, Denjun, did a meta-analysis of 500 peer-reviewed papers from this time period and almost 37,000 patents, specifically focused on nanopesticides. Now, one of the interesting things is most of these authors actually work for the U.S. Environmental Protection Agency. So I think this is addressing that, starting to address that previous issue about regulatory concerns. So from this meta-analysis, what Kevin showed, Denjun, was that on average, you get a 31% increase in efficacy when you use nanopesticides. Remember, efficacy is kind of a function of, the amount applied and the effect that you get. And that included almost 19% increased efficacy in field studies. So again, in 3 years, we've accumulated more field data. But what's also more interesting to me as an environmental toxicologist are some of these other benefits. So when you use a pesticide in nanoscale form, not only do you get that 19 to 35% benefit or whatever the numbers were, but you actually get 43% reduced toxicity to non-target organisms. And you decrease the loss of your active ingredient. So you're getting these upstream benefits, but you're also getting these downstream benefits of less impact on the environment. And again, the fact that this is from a group of authors, many of whom will work for the EPA, I think is significant because it's demonstrating that really, I think there's a lot of potential in this field. So one other thing that I wanted to focus on goes back to where I started, that my training is environmental toxicology. This has become a really active area of research for us, probably 60 or so papers in the last 6 years, including a bunch of additional grants 2 years ago and last year. We've talked about all of the mechanisms, but what I wanted to focus on is this concept of safety and sustainability. 3 of these studies, what we actually showed is that the strategy that was being used, whatever it was conveying some benefit to the plant. But what we also saw, because of the endpoints we measured and because we're concerned about safety and sustainability, was that we were getting some unintended negative consequences, either in the rhizosphere or with endophytes. We would argue that if you're seeing these negative consequences, even if they're not the primary endpoint, then the strategy that you're looking at in terms of safety and sustainability is probably not something worth pursuing. So it's really, really important to keep these concepts in mind as we move forward, because there's a lot of people moving into this space, and I'm starting to get concerned that there's a lot of work that's going to happen that's going to focus on strategies that really are never going to work under the field scale because they're too expensive or there's just going to be safety or sustainability concerns. So just to conclude, hopefully I've convinced you at least partially that nanotechnology really has tremendous potential here but not only in agriculture, but also to really address this issue of global food insecurity, which is becoming more intractable more quickly than we thought it was going to be. There's lots of different ways along the crop production chain to impact these. There are topics we didn't talk about as well, like food preservation and packaging materials. But the key concept here is safety and sustainability for us. And to understand if something's safe and sustainable, you have to understand what the mechanisms of action are. Obviously, I have a very large number of collaborators, both in the U.S. and in other countries, and I'm always looking to expand that list of collaborators. So if there's something here that kind of struck you as interesting, please feel free to reach out. There's a lot of interest in this work. These documents are they come out of the White House. They're supplemental budget requests from 2019 and 2020 for the National Nanotechnology Initiative, which is how the US government funds nanotechnology research across 26 agencies. And in these White House budget requests for 2019 and 2020, they actually, our work was featured, which again was really exciting for us. But I know I went 5 minutes longer than I was supposed to, so I will stop now and hopefully entertain some questions.